Welcome to Virology Live. This is session number 12. Today, we're going to talk about infection basics. This is the launch of the second half of this course where we're going to learn all about infection of hosts, mostly humans. It's a human-centric virology course for sure. And today, I want to go through some of the basic principles for understanding how viruses infect human hosts. So far in this course, we have talked about, I got to get my font back, my mouse back. Where is it here? Sorry about that. Slideshow mouse, where is the damn thing? Show pointer, here we go, all the time. All right, sorry. So far in this course, we have talked about reproduction of viruses in cells in culture. And from now on, we're going to be talking about reproduction in hosts, as I've already told you. But in both cases, there are some commonalities, of course. In both the infected cell and the infected host, viruses must get in. They must get into cells in culture. They must get into the host. They must reproduce, and they have to get out. Now, in cells in culture, getting out is not crucial, right? Because that's the end of the line for those viruses. But in a host, getting out and finding another host is crucial. And that's the nature of host-parasite interactions. The viral genome has to establish itself in a host population to endure. In the cells in the lab, it doesn't have to, because the investigator is always there to put them back in. But in nature, viruses need to find a new host. And of course, they're not finding anything. They are bumping into new hosts. And that's one of the reasons why virus reproduction makes very large numbers of progeny. As I said on day one, we live and prosper in a cloud of viruses. Most virus encounters have no consequence. You are bombarded on a daily basis by many different viruses, and you're fine. And yes, you are. It doesn't matter who you are. You are bombarded on a daily basis by all sorts of viruses. Many infections never get established for reasons we talked about already. Many infections are inapparent or asymptomatic. Sort of the same word, although I would prefer asymptomatic, because it has a very specific meaning. And now we need to define signs and symptoms. If you don't already know this, a sign is evidence of disease that can be observed by others. So if you have a rash, other people can observe that, caused by chickenpox or measles or varicella zoster virus or measles virus. But if you'd have to take blood from someone. You can measure virus levels in the blood. You can measure cytokines. You can measure B cells, T cells, antibodies. Those are signs of infection that other people can observe. A symptom is what only you can feel. It's apparent only to the patient. If you have an upset stomach, if you have a headache, a fever, I guess a fever could be measured, malaise, muscle aches, brain fog, or anything of that sort, it's evident only to you. That is a symptom. And that is where the word asymptomatic comes from. It's very important. It means you don't feel anything, but you certainly have signs of infection if people measured it. If a virus is in you and reproducing, you may not have symptoms, but you're going to have signs. You're going to have viral loads increasing. You're going to have an immune response, etc. Okay, very important distinction. And that's the way to understand an asymptomatic infection. Many people tell me, well, how could it be asymptomatic if the virus is reproducing? Well, there are signs. They're just not symptoms. So here's an example of this, West Nile virus. Absent from the U.S., in fact, the Western Hemisphere, before 1999. It arrived in New York City. Great story, by the way. Go read West Nile story by Dixon Despommier. Entered the U.S. in the summer of 1999. This is a flavivirus transmitted by culicine mosquitoes. And by October 2004, it had spread across the country. About 1 million people were infected. How do we know this? We do serological surveys. We measure antibodies in people. About 20% of these infected people developed febrile illness. How do we know that? Again, serological surveys, records of illness in some patients where you ask them. It's not a precise science, but it's the best we can do. And central nervous system illness developed in about 1% of infected people. This is the rate of CNS complications for West Nile virus. So febrile illness developed in about 20% of infected people. Many people were infected with no obvious disease to them, no symptoms, although there were signs. Again, serological, seroconversion, and other things you can measure. And that is what an asymptomatic infection is. And this is a problem for many viruses because if you have a lot of asymptomatic infections, it's hard to stop an outbreak. Witness SARS-CoV-2, right? Many asymptomatic infections. Contrast it with SARS-CoV, number 1 from 2003. We'll talk about that later today. Here's the spread of West Nile virus throughout the U.S. Entered New York City in the summer of '99 and then slowly spread throughout the U.S. The yellow are low numbers of cases, the red are higher numbers of cases, until eventually it was across the entire country, Canada, Southern Hemisphere, etc. The virus had originally been identified in 1947 in the West Nile district of Uganda. Another example of asymptomatic infections, SARS-CoV-2. Here is just a overview of what happens, different kinds of disease. So we have asymptomatic disease. Here's one of my font problems that I didn't solve. Yeah, let's fix that. Good old, still doesn't look very good. Wow. This is supposed to be Avenir book. Why isn't it? It's because I selected the wrong thing. There we go. Got to know that 20% of cases are asymptomatic. Asymptomatic and mild, 80% overall, so that's a combination of asymptomatic and mild. Mild, we mean fever, fatigue, dry cough, ground glass, opacities in your lung, which you can see on an imaging, pneumonia. These can be mild. Maybe you wouldn't call pneumonia mild, but it can be. And then 14% severe disease and critical and deceased 5%. But the key here is that 20% asymptomatic. These people don't feel anything. But if you measure them, you can get signs of infection. That's what I want you to get out of this. And that leads us to the incubation period. This is the initial period before symptoms of disease are obvious. Symptoms, not signs. And words are important, folks. Signs are obviously present. You're infected. You get infected. Here's a timeline of this incubation period. You get infected. The viruses enter, say, your upper tract. They enter cells. They begin to reproduce, and the cells produce more and more virus. So you could measure this increase in virus. You could measure viral genomes replicating. You could measure the response of the host very early on in terms of interferons and other cytokines. And then that is the period of having signs, but no symptoms. You're asymptomatic. And then the onset of disease, that's the symptomatic period, and eventually resolution. So the incubation period is before you see symptoms of disease, okay? Symptoms, what you can feel. Very specific definition. Now, depending on the virus, it may or may not be transmitted to others during the incubation period. Not all viruses are transmitted during the incubation period. For example, Ebola viruses, not transmitted until you have symptoms. However, we know that SARS-CoV-2 is, so it depends on the virus. All right, here are incubation periods of some viral infections, and you can see they range from 1 to 2 days for influenza viruses, 1 to 3 days for rhinoviruses. Some of them have a longer range, 2 to 21 days for Ebola viruses, all the way up to 50 to 150 days for papillomaviruses, which cause warts. Here's SARS-CoV-2, which, it emerged just as we were completing the 5th edition of the textbook, so it's not in this table. 1 to 14 days incubation period. And in general, we think viruses with short incubation periods, their reproduction at the primary site of entry is what produces the symptoms. And then the ones with long incubation periods, the viruses need to spread within the host, and the symptoms are caused beyond the primary site. And a great example is measles virus, which enters the respiratory tract. And then the rash, the virus needs to reach your skin. It does so through the blood, so it takes time to get there. Another word that you may hear is prodrome. I'm not going to use it anymore, but just in case you see it in the literature, it's the period of symptoms before those characteristic of the disease. It's from a Greek word prodromos, meaning precursor. And what does that mean? Well, characteristic of disease, for example, the rash of measles virus. Before that, you might have fever, malaise, etc. Those are not characteristic of the disease. Those are general symptoms that can be seen with almost any virus infection. So the characteristic symptoms of disease, that is the prodrome. It's a little bit different from incubation period. Now, a few other terms that we need to talk about here, shown on this chart: morbidity, mortality, incidence, and fatality. And this is a figure from Principles of Virology where each icon is 10,000 people. The blue people are healthy. If the infected people, PCR positive people, are orange, and then people with signs, symptoms are red. And then you know what the coffin means, right? That's how many people have died. So here, 10,000 people have died. And so incidence is shown in the first panel, morbidity in the second, mortality in the third. And I actually have some other definitions that are not illustrated here. Incidence, just the number of people infected over the number in the population with time. There's a time component to that. Morbidity is the number of people ill over the number in the population. So that you can see infected versus ill is different. The mortality rate is the number of deaths over the number in the population. Case fatality rate, the number of deaths over the number of confirmed infected, by PCR, so forth. CFR, so 10,000 deaths in 50,000 infections, that would be 25%. And that was a number bandied about extensively at the beginning of the SARS-CoV-2 outbreak. I'll get to that in a moment. The real calculator of death is the infection fatality rate, the number of deaths over the number of actual infections. And you will recognize right away that it's very hard to know the number of actual infections because we don't test everyone. So it is usually statistically calculated from the number of infections that we detect by PCR. And that's typically been less than 1% for SARS-CoV-2. Now, the CFR has been higher, but I'll get to that in a moment. Another number which we talk about in terms of infection is the basic reproduction number, R naught. And R naught is calculated by this formula. R naught equals tau times C times D. And tau is the probability of infection given contact. C is the average duration of contact between the infected and the uninfected host. And D is the duration of infectivity. Now, you can see that this is a combination of viral properties and human behavior. And so R naught is a squishy number. I would not look at it as a constant, even though we do treat it as such, and I'll show you in a moment an image that does that. But it's not. It's squishy. It's hard to calculate. Human behavior is hard to quantify. The duration of infectivity is a virus property, of course, how long you're shedding enough virus to infect another person. But the average duration of contact, that's how much time people spend together, the density of people and so forth. And then the probability of infection given contact is, I also think, influenced by viral properties. Now, what does this number mean? Well, it's the number of secondary infections that can arise in a population of susceptible hosts from a single infected individual. So I'm infected. How many people can I infect? But it's an average. It's an average throughout the population. It's typically, R naught is typically calculated early in an outbreak before interventions or population immunity kick in. And so if you hear people today talking about the R naught of Delta, they're wrong. You can't do an R naught for Delta. You have to do something else. You have to do an estimated, an RE, or I forgot what T stands for. Someone put it in there. But it's RE or RT. Because you've already got interventions, you've already got immunity, you can't calculate R naught. But even R naught at the beginning is squishy, as I said. Now, what does it mean? If it's less than 1, the epidemic will not be sustained. If it's greater than 1, an epidemic is possible. If it's much greater than an epidemic is certain. Early in infectant SARS-CoV-2, R naught was calculated between 2 and 3. I don't see any evidence that it's any different. Well, the R naught is the same. RET varies according to population behavior, according to immunity. Is it changed by the virus? I don't see the evidence. A lot of people will tell you I'm wrong. Hey, doesn't matter. As you see, the time of contact between individuals, the length of the infectious period can influence R naught, and interventions, of course. So you can't do an R naught after the earliest days of an outbreak. So let's compare CFR, case fatality ratio, and R naught. This is a figure from the New York Times. And again, this is like saying these viruses have a fixed R naught. But even, and again, the definition is you can only determine the R naught early in an outbreak, before population immunity, before interventions. So presumably that's what this is. And then we have case fatality ratio. So here we have SARS-CoV-2, which is CFR is going from 0.1 to 3, but I would say that really depends on many factors as well. I'm not sure you could give it a range like this. And then the R naught, 2 to 3 for SARS-CoV-2. You can see some other viruses here. Let's take a look at R naughts. Quite low for seasonal flu, the 2009 pandemic influenza, common cold viruses like rhinovirus, poliovirus. Then we get into the higher ones, the serious players here, chickenpox and measles virus, the highest as far as we know. 15, one person can infect on average 15 other people. Here's smallpox. Now, other viruses are shown up here just to show you that the numbers among the different influenza viruses can vary. And we have MERS and SARS up here with a relatively high CFR, but again, an R naught in the same range of SARS-CoV-2. All right, so those, you have to take these with a grain of salt. You have to assume that the R naught was calculated properly at the beginning of an outbreak. It's very hard to do. Now, CFR, one size does not fit all. And this, at the beginning of the outbreak, many people were saying, oh, the CFR is so high for this virus, it's lethal. Well, it depends where you look. And let me show you some data. Here's the first 44,000 cases out of China. This is data from the WHO China Joint Mission published early in 2020. 44,000 cases, 1,000 deaths, case fatality ratio 2.3%. It's a simple math. This divided this into this, right? However, if you stratify by age, you get a different picture. No deaths in the 0 to 9 years, then slightly increasing as we go up, but then you go over 80, 15% CFR. So you see age affects CFR. So does sex. Men about twice the CFR as women in this initial outbreak. And you never should say this is what it is, because it will vary elsewhere. Look at the graph on the right. This is the CFR on the y-axis with time period January to 20th February 2020. And look at Wuhan, the blue line. In the beginning of January, CFR is 20-some-odd%. And then it goes down to less than 5% by February. Why? Did the virus change? Absolutely not. Some people would say it did. And if the curve went up, they would definitely say the virus did. But you have to look elsewhere before you make conclusions. What happened here? Well, initially, Wuhan hospitals were overwhelmed. And then they figured out how to deal with it. And they also figured out how to deal with patients. If you look outside of Wuhan, Hubei, the larger province, much lower CFR from the beginning. China outside of Hubei. Hubei, where Wuhan is. Hubei, outside of Hubei in all of China, also low. Why? Fewer cases, hospitals can handle it. And so it just shows you that CFR isn't just a property of the virus. It's how we respond to it. I got another font issue here. Let's fix that. I missed all these. I went through. China CDC Weekly. Okay. The other property I want to mention very briefly, overdispersion parameter, which is represented by K. And if you remember, I remember this very clearly. In some countries, the importation of the virus was associated with fewer secondary cases than you would expect with a virus with an R naught of 2 to 3. I remember seeing in the first months, lots of cases in China and hardly anything elsewhere, yet the virus was there. And this is part of the reason, of course, it's also testing. And this observation suggested that not all symptomatic cases cause transmission, which is also observed for SARS-CoV. And that is calculated by overdispersion. That is high-level individual variation in the distribution of the number of secondary transmissions. And that's calculated as K. For SARS-CoV-2, K is 0.1, which means, and this is amazing, 80% of transmissions are caused by 10% of infectious individuals. Why is that? They make the most virus. In a study out of the University of Colorado Boulder that we covered on TWIV some time ago, they did quantification of PCR levels for all of the positives. And they found that 10% of the positives had 90% of the viral loads. So a few people can reproduce virus to very high levels. Why that is, we don't know. And those are the ones that transmit. And this is why if you have a family, my brother was positive, but none of the other 5 family members got infected. This is why. Because of the overdispersion parameter. Everything can be explained eventually. And what that means is we get super spreader events like this one in Hong Kong, reported in Nature Medicine, which we also covered on TWIV. These on the right here, panel D, these are chains of infection. One person infects 2 or 3 or 4. These are not super spreader events. But here on A, this is a super spreader event. This one individual, B, infected a lot of other people who then infected a lot of other people and amplified the outbreak. And these kinds of super spreader events involve someone who is highly transmissible, has a lot of virus, and some venue that allows for extensive transmission, like social venues, bars, restaurants, before all the lockdowns, churches, funerals, etc., things like that. Okay? All right, let's take a little quiz here. All right. My thingy, I have another parameter to set to be able to do that. Okay. Socrative. Here we are. Which of the following parameters is not influenced by human interventions? Mortality rate, case fatality ratio, reproductive index, incidence, incubation period. All right, while you're looking at that, let's check out some questions here. So you like my little improvement here? I got a shelf, and I'm going to put stuff on it, little viruses. Fields Virology, there's Greg Zuckerman's book, A Shot to Save the World. And I'll populate it if you like this look. Okay, let us see what we have here. Today's session, we will get to understand more how to judge stories in the wild. You bet. From today on, actually. But today is a big one, right? A lot of discussion of boosters, but we're not going to do that now. We're going to wait for the vaccine session. And everyone knows the exam is up. The midterm exam went up Monday. It took me hours to post it on SurveyMonkey because I had to copy paste all my questions, and then anything over 10 in SurveyMonkey you have to pay for. So it's there. So go take it. Yeah, you can change the font master, but there are individual things that you put in that don't respond to master, so you still have to do those. That's okay. Thank you, Marge, for your contribution. Really appreciate it. Coughing, sneezing, sweating. Well, a cough is a sign because other people can see it. Sneezing is a sign. Other people can see it. A symptom is what only you can feel. Oh, this is cool. The symptoma is chance in Greek. I don't know why that would be. Yes, and that was answered, so sorry about that. I don't mean to usurp you. Yes, take the midterm, folks. I put a lot of work into it. Take it. And take last week's quiz as well. I don't know how long the midterm will be up. I don't want to have it up forever, okay? How is tau determined? That's a very good question, actually. Tau is the probability of infection given contact. I really need to look into that because it's important. It's determined by the length of time that people are together. Well, that's C, right? But I think it's got to be influenced by the length of time and how much virus is shed, right? Because you need a certain amount of virus. But actually how it's done, I must admit, I've never looked into. Maybe we need to have a little session on that. So RE is the growth rate. Yeah, and that's wrong because you have to take into account what people are doing. And if so, you calculate RE and you find it's 5 for SARS-CoV-2, then people say, oh, the variant is more transmissible. That's wrong conclusion. It's just movement of the virus, or as you say, at growth rate, and that's determined by viral fitness, as we're going to talk about. Overdispersion is also hard to measure. Look, epidemiological parameters are hard to measure. Most of the time they are statistical calculations. It's not what I would call observational data or data generated by an experiment. Not to say that it's bad. I'm just saying it's not easy. Thank you, Marianne, for your contribution. Really appreciate it. Background needs to be darker. Why does the background need to be darker? It can be whatever I want. I mean, I appreciate your suggestions, but I see many videos with light backgrounds. Anyway, there's going to be a watercolor here eventually. I told you, this is a work in progress. I shouldn't show it to you until I'm done. You missed the phage cushion. I do too. It's not going to stay in my office forever, but where would I put it? If I put it on the shelf, that would be kind of weird, a cushion on a shelf, right? And there's no couch here in the incubator, so no, there are 42 questions on the midterm. I paid so you could have a great midterm, okay? It's like $26 a month for SurveyMonkey. Now, I'm not complaining. You guys support us very nicely, but there are 42 questions. And where is the midterm? Go to the syllabus. Someone will certainly have put in the chat here. Okay. There's no time limit on the midterm. And I don't know if you can actually stop and go back. You have to ask someone who took it. How do you measure the development of herd immunity in a local population? Well, that's done by serological assays, really. Listen, folks, don't worry. Just take it. I mean, nobody else sees your grade except you. Now, you're your own biggest critic. I understand that. But just take it. It's part of the learning process. The syllabus is virology.ws/virology-live-2021. I know you kept repeating this. Why are there no data generated to determine the transmissibility? Well, it's hard to do, right? You can't do a transmission experiment in people. So you need to do lab experiments that are really surrogates, like study how quickly the virus reproduces compared to ancestral strains, what it does in an animal model. Those are being done, and we'll see over the next year what we get. If the data suggests that it could be more transmissible, I will revise my position. But I don't want conclusions drawn until we have the data. I need a 3D printed phage also. Oh, I think I brought one. Where is it? I had a pin. I meant to leave it on my desk. I need a big one. I agree. I'm going to bring my 3D printed polio and SARS-CoV-2. Yeah. I don't know where I'm going to put them. They don't have a lot of space. The shelf is really big, but should I move it all behind me? I don't know. Give me some design advice. Okay, let's see what we did on the quiz now. Let's get back to that. The incubation period is not influenced by human interventions. That's right. Everything else is. These are all. I mean, some people said a reproductive index is not. It is. That was one of my main points here. All these things are, except for incubation period. All right. Okay. Let's talk about viral pathogenesis now. You've got your primer on some of the basic parameters of transmission and death and so forth. Let's talk about how viruses cause disease. Viral pathogenesis, the process of producing a disease. It could be viral pathogenesis, could be bacterial, fungal, could be pathogenesis caused by a car accident or by stress. We talk about viral pathogenesis, how viruses cause disease. There are always two components of viral disease. Don't forget this. If you could only remember one thing, remember this. There are the effects of viral replication on the host, but there are effects of the host response on the virus and the host. And in many cases, the virus actually causes no damage, and it's entirely the immune response of the host that causes the signs and symptoms of the disease. We'll talk about that in another session. All right, two components of pathogenesis, what the virus does and what the immune response does. Immune response is a two-sided, double-edged sword. All right, fundamental questions of viral pathogenesis. Why do I have this delay between me switching and me seeing it on? Am I frozen? No, I'm not. It's the story here, computer. Why do we always have to have some issue? Oh, look at that. Isn't that great? No, don't like that. Let me plug it into another port. Maybe I should go back to chalk and erasers. Oh my gosh. There we go. All right, these are the questions that we ask when we study viral pathogenesis. How does the virus enter the host? Clearly. What's the initial host response? Where does the virus first reproduce? That is, where's primary replication? How does the virus spread? what organs and tissue are infected. How does the host respond in terms of antibodies, interferons, T cells, etc.? Is the infection cleared, or is a persistent infection established? We'll see many examples of that. And then how is the virus transmitted to other hosts? So we'll address some of these questions in this course. There are 3 requirements for a successful infection of a host. First, you need enough virus. You know, if you inhale 1 PFU, 1 plaque-forming unit of a virus, is not likely to infect you. And how much you need depends on the virus. We don't know how much you need for SARS-CoV-2. We do know for some viruses because we can do challenge experiments. So something like 10 particles of a norovirus are enough to infect people because we can do human challenge experiments. All right, you need enough virus. Where that virus goes, the cells have to be accessible, they have to be susceptible, and they have to be permissive. Accessible is clear, right? If the virus can't get to them, they're not going to be infected. And you know what susceptible and permissive means. We've already learned that. And finally, local antiviral defenses either have to be absent or overcome to allow that first infection. So if I inhale some virus and I don't have any antibodies in my upper tract, the virus is going to start reproducing until maybe the memory response kicks in, the memory to a vaccine or another infection with a similar virus. The site of entry is critical, of course, and in fact, our human body only has a limited spectrum of entry sites for viral infection. Yeah, they're limited, and they're obvious, and they're shown on this picture. We have the skin. We have the respiratory tract. We have the alimentary tract, the urogenital tract, and we have the conjunctiva, the eye. I would lump that with mucosal surfaces like respiratory mucosa and GI mucosa and urogenital mucosa. So there are not that many places where viruses can get in. It's good. We're well protected. Let's start with the skin. Biggest organ in the body. Outer layer is dead, though. Here's a cross-section of skin. This outer layer. Well, the outermost layer is called the stratum corneum. Those cells are all dead. That's what flakes off, makes a lot of the dust that you see when sun shines through the window. And then below it, now we have some living cells. And those 2 layers constitute the epidermis. So viruses landing on the skin will not reproduce because the skin is dead. The outer layer is dead. That's a great barrier, right? Put up dead cells so that viruses won't reproduce. However, it's not hard for viruses to get past that. Below the epidermis is the basement membrane, which is as we've discussed before, it's a very substantial layer of glycoproteins, glycolipids, and other highly polymerized proteins. That's a barrier to viruses just passing through. And below that is the dermis, where you have blood vessels and lymphatic vessels, hair follicles, you have lymph cells, you have macrophages, and then getting into that is where viruses can take a ride in the bloodstream. Now, there are many ways to breach this lovely skin. Of course, mosquitoes do that very well. The female mosquitoes, in order to lay eggs, need to have a blood meal, and so they are looking for blood vessels. Their proboscis inserts into the dermis. They actually probe and look for blood vessels. And as they're doing the probing, they are secreting things out of their proboscis. Saliva, which contains chemicals that anesthetize you so you don't slap the mosquito. They contain anticoagulants. As Dixon says, it's a veritable pharmacopoeia coming out of the mosquito saliva. And, of course, if the mosquito had been infected from a previous blood meal, that mosquito is putting viruses into you. The mosquito actually inserts its proboscis into a blood vessel. Amazing. And this video, go check this out. There is a video there taken by researchers of a mouse skin from underneath, and you can see the mosquito probing and probing and probing, and then it hits the vessel, and it sucks, and the vessel collapses from the suction. Oh my gosh, it's just amazing. Go check that out. So mosquitoes put viruses through the skin, obviously, right into your blood. All right, you can obviously get scratches. You can get injuries of various sorts, dog bites or animal bites, they can breach the skin as well. So let's take up mucosal surfaces now. These are great for virus infection because they're lined by living cells, right? Well, the eye, the mouth, the respiratory tract, the GI tract, urogenital tract, they have to communicate with the outside world. So they're lined with living cells. So those are absolutely exposed to the world for viruses to infect them. So the upper tract, of course, with the respiratory tract, I should say, in general, is lined with respiratory epithelial cells, shown here. They have typically a brush border. Here's some goblet cells. So it's a complicated epithelial sheet. It's not just made up of one kind of cell. There are multiple cell types. Here's a goblet cell that makes mucus. Mucus lines the apical border of the respiratory epithelium and serves as a primary physical defense, as we'll see next week. And then there's a basement membrane, which prevents things from just floating across from the apical layer, like viruses. And this kind of layer lines most of the respiratory tract. There are some differences down in the very lower parts of the lung in terms of the cells that are present, but they're still epithelial cells. And so down as we move past the upper respiratory tract, the nasopharynx, back of the mouth, and we reach the trachea, the bronchi, the bronchioles, and eventually the alveoli. And as we move down into the trachea, the epithelial cells are ciliated, and they're constantly beating. This is called the mucociliary elevator, because as virus particles move down into the tract, they get caught in the mucus, and there's this constant beating, which brings the mucus up into your mouth. You then swallow it and digest the virus. Yes, when you were doing the swallowing, you've got a little bit of mucus in there coming up from your tract. And of course, if you're infected, there's more mucus produced, so it's more substantial. But that's another physical defense for you to swallow your mucus. Mucociliary elevator. Now, viruses infect the entirety of the respiratory tract because these cells are exposed to the air. You inhale them, they can cause upper respiratory tract infections. Many viruses are restricted to the upper tract and cause syndromes like the common cold or rhinitis, pharyngitis, inflammation of the pharynx, laryngitis, inflammation of the larynx. Many viruses, including these same ones, can go into the lower tract and cause more severe disease. When you start to reproduce in the alveoli, for example, that compromises oxygen uptake in a variety of ways. And these are some of the viruses that can cause lower tract infections, which we also give names like tracheitis, bronchitis, bronchiolitis, and bronchopneumonia, reproduction in the very lower reaches of the respiratory tract. The alimentary tract is also fair game for reproduction. It's also lined with epithelial cells, as we'll see. Typically, the small and the large intestines are sites of entry. And remember, the stomach is highly acidic, and so many viruses are immediately inactivated in the stomach. And by the way, this idea of swallowing your mucus, so if you have a respiratory tract infection and you're swallowing mucus with viruses in it, where do you think those viruses are going to end up? Yeah, they're going to be in your feces. Do you think most of them are infectious? No, they're just being pumped through your tract to get rid of them. So finding viral nucleic acid by PCR in feces does not mean they're infecting your GI tract. So many people assume that. Okay, let's take a look at the alimentary tract. I was just saying the stomach is highly acidic, so many viruses are inactivated. The small intestine is alkaline, and plus there are bile salts and proteases and so forth. It's a pretty hostile environment, yet a number of viruses make it through and infect the epithelial cells. Here's a cross-section of the small intestine. It's, again, lined with epithelial cells. There's a basement membrane below them, and then there are below the basement membrane what we call the subepithelial tissues, where we have blood vessels, lymph vessels. In terms of the small intestine, there's muscle cells and so forth. Now, there are also different kinds of cells within the intestinal tract besides the epithelial cells, which are called enterocytes, to tell you that this is the gut we're talking about. Here, for example, is an M cell. M stands for microfold. It is a kind of cell that is very thin and is involved in sampling the contents of the lumen of your intestine. And so what happens is these cells are actively transcytosing. What does that mean? Materials bind to the apical domain of the M cell. They're taken up by endocytosis and then deposited on the basolateral side. And there are immune cells that can sample it. There are lymphocytes and macrophages that you can see are snuggling up very closely to the basolateral domain of this M cell. So they sample and say, is there anything foreign here that we should be concerned about? And how that sampling works is that these cells will take a sample and bring it back to a local lymph node, and the T cells there will help them out. We'll see that next week. So here's an EM, by the way, of an M cell. So you can see two enterocytes on either side with their cilia or microvilli. And then the M cell is quite thin. And here's some lymphocytes underneath it. And this is actually taken from a mouse infected with reovirus, which are these small dots. And reovirus is actually taken up through M cells. It takes a ride on the process by which the M cells transcytose material into the subepithelial spaces. So here are some buzzwords about the small intestine. A selectively permeable barrier lets some things in but not others. It comprises polarized epithelial cells. And of course, these have direct contact with the outside world because that's what the lumen of your gut is, right? It's open on both ends to the outside world. So you may not think so, but that's how we view it, virologists anyway. And this intestine has direct contact with the immune system, as I've told you, and it's also highly innervated, which we don't see here, but there are nerves innervating. There's an enteric nervous system that connects the gut with the brain. Your genital tract is another mucosal area lined with mucosal epithelial cells in a similar structure to what we've talked about. The urogenital tract is also protected by mucus. It also is protected by low pH. It can be sites of virus entry. Minute abrasions from sexual activity can allow viruses to enter. Some viruses produce local lesions like warts, human papillomaviruses, and of certain types of human papillomaviruses, the warts can proceed to cancers, as we'll talk about later. And other viruses enter in the urogenital tract and spread elsewhere, like HIV and herpes simplex viruses. So another portal of entry, and we'll talk about that when we talk about some of these specific viruses. The eye is another place where you can get a virus infection. If you've ever had a red bleed in your eye, it's called a subconjunctival bleed. This can be caused by a virus infection. I get one about once every 2 months, and it's not caused by a virus infection. I just had one last week, in fact. And I've discussed this with my ophthalmologist every time, and she says, nothing to do. I don't know why they happen. But viruses can cause this also. This is rupture of the capillaries and bleeding. It looks very shocking, but most of the time the virus infections are contained, but sometimes they can spread elsewhere, which is not good. Anyway, here's a schematic of the eye and showing you the different cell types that can be infected with viruses. For example, the conjunctival epithelial cells is this very thin layer of cells that actually goes from beneath the eyelid over the front of the eye to the other eyelid. Those can be infected with viruses that you can see here. The corneal fibroblasts can be infected, either the stromal fibroblasts or that's the cells that make up the body of the cornea, or the epithelium covering the cornea can be infected. Even retinal pigment cells can be infected by influenza virus. So another portal of entry, if you swim in, say, pools or hot tubs that are improperly sanitized, there may be viruses in them, and you often can get adenovirus infections. They're often associated with improperly sanitized pools. The fetus is another site of infection that we should talk about. The developing fetus is highly influenced by the mother, of course. There is a placenta that separates the fetal from the maternal circulation, but some viruses are able to cross the placenta. We call that transplacental infection. And that is typically caused by what we call the torch pathogens. That acronym stands for toxoplasma, which of course is not a virus, but it is a torch pathogen. Rubella virus, cytomegalovirus, HIV. And the O was reserved for other because it was wisely understood that we wouldn't get them all. And in fact, Zika virus was our most recent addition to the torch pathogens. Just, a virus that we've known since 1940s. And suddenly in 2015, it's associated with birth defects, congenital birth defects, and it turns out to be able to cross the placenta. So here we have Zika virus. So if the mother is infected, which can happen by mosquito bites, the mother has virus in the blood, and the virus is able to cross into the fetal circulation and infect fetal tissues and cause problems. And rubella virus does that, and cytomegalovirus, HIV as well. So that's transplacental infection. You also have what we call perinatal infection, which means at birth, right? There's a lot of blood present at birth, and if the blood has virus in it, then that can infect the fetus. And so some viruses do both. HIV can infect transplacentally or perinatally. All right, there we go. I can do my switching right from within Keynote. That is a preference. The outer layer of which of the following is dead but can still serve as a portal of virus entry? Respiratory tract, alimentary tract, eye, skin, urogenital tract. This has got to be 100%, folks. You have to get 100 on this. No pressure. All right, let's go back to the questions. Thank you, Kathy, for your contribution. Oh, you like the midterm? Good. I hope you like it. Those are the same questions that I give my students. So they're tough. They're not easy questions. And they're mostly multiple choice or multiple answers, so be careful. But, multiple choice is a circle and multiple answers is a box. You can check as many as you think are right. Those are the hard ones. Multiple choice is kind of easy because there's only one, and most of the time you could guess it. You think you can move the shelf to the middle while I don't have the watercolor? No, Michael Min is not the best ever background. I'm sorry. No. My background is the best ever, wherever it may be. Yeah, I know he has a nice bookshelf, but it's way too cluttered. Way too cluttered. It's not the best ever. I want to put this watercolor, which I have. Here, let me show you the watercolor. I can do that very readily. Let's just hide some of this stuff. You're going to love this. Look at this. Look at this watercolor. That's what I had commissioned by Michelle Banks. You can see her signature down here on the right. And I said, take the Big Sur palette. The Big Sur was the previous operating system of Macintosh. Take that color palette, which I really liked, and make a watercolor. So she made a really big one. And wow, isn't that cool? I love it. All right, back to the questions. I had given up in these classes. Oh, man, why would you give up? Didn't Winston Churchill say, never, ever, 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 ever give up? I'm not criticizing you, but don't give up. Giving up can be infectious. You give up one thing, and you're going to give up something else that is really important, right? Is gastric mucosa phlegm carried by cells to an infected area? I know phlegm is viral, and immune debris could gastric phlegm is just put through the whole intestinal tract, and it's in the feces. Your feces has a high content of phlegm. Mathematicians would call the gut outside topologically. Yeah, it is outside. Why doesn't the immune system react to food cells? Well, sometimes it does, right? And but it learns what is there. I think, and that's part of it. Do epithelial cells present receptors? Absolutely, yeah. That's how viruses can infect epithelial cells. They have receptors on the apical surface. Would viruses infecting the eye also reproduce? Yes. In many cases, not all, they don't have to, but yes, they often do. Thank you, John, for your contribution. Glad you like it. Then, yes, you listened. The placenta would not exist without HERVs. That's right. The fusion of that outer layer was caused by a glycoprotein from a HERV. Yep. Let me just say another thing about Michael Minna while you guys are praising him. He became famous on TWIV, okay? Period. He's got incredible exposure from TWIV, and he'll tell you that. And yes, it's the content that matters. But I'm very sensitive about my background. So to me, just don't say that, okay? But you can say it elsewhere. What prevents the ear from being infected? Actually, most of the time when your kids have an ear infection, it's viral. And for years, we used to treat them with antibiotics. Well, our kids, all three kids, had constant ear infections. They always got antibiotics. They were mostly viral. And that was eventually recognized, which you can't treat, of course. Does the virus have the same cycle when it's in different cells? Well, that's a good question. I think we should distinguish between productive and non-productive because many cells are not permissive, right? And so it's going to stop at some point, and that's going to be the different point. Why do some viruses cross the placenta and others do not? I think we have zero understanding of that. In fact, they may be carried in exosomes. They may be brought over in other ways. I'm not aware of a good explanation for that. I'm just kidding about Minna. Well, not so much, but I know it'll be very cool. But I know, in fact, on TWIV, we said Minna's background was very cool. That's fine. I do like my phage pillow, though. It's all right. Yes. Thank you, Rob, for your contribution. Did Minna overpromise? I think so. And Amy has talked about that a lot. He promised when we didn't have antibodies that could be used for rapid tests. Now we have some rapid antigen tests, but they're not cheap, right? Are there any data as an eye is a site of infection? I have not seen any data saying that the eye is a site of infection. Most of the reproduction reports outside of the respiratory tract are suspect in my view. And don't worry, Cherry, you didn't offend me at all. I'm just joking. We have a background competition, right? That's fine. Minna's is very nice. Why viruses prevent one another from doubly infecting? So you can actually have multiple infections. We're beginning to realize that more. It's becoming a science, and we're realizing that the doubly infected person has a different disease from the singly infected person, right? However, viruses will prevent their own kind from infecting. Many viruses, when they infect a cell, will downregulate receptors on the surface, and we think that is for preventing more viruses to infect. I mean, that would be in the case of a long-term infection, right? Not one where you're going to kill the cell right away. That wouldn't make much sense, right? This is an interesting series of events. I would never have heard of Vincent or Michael Minna if it weren't for Lex Friedman. I would never have heard of Lex Friedman if it wasn't for Joe Rogan. So whatever you think of Rogan, I guess the outcome could be good, right? All right, let's move back to our quiz here. We have to go back to me. And where is the super source? Oh, I see. I see what I've done wrong. Sorry. Infection basics. Show the results. All right. 98%. 2% of you said the eye. The outer layer of the eye is not dead. It's that conjunctival layer. Those are living cells. But that's probably one person out of 62, right? Okay. Viral spread. So we've got a virus in you. Now, how does it go elsewhere? Where does it go? Some viruses don't go anywhere. They remain localized. So a virus here infects this respiratory epithelium. It is shed maybe laterally, infects neighboring cells. It could spread throughout the respiratory epithelium and just remains there and causes a common cold. The virus spreads within the epithelium, is contained by the tissue structure and the immune system. But other viruses spread beyond the primary site. They are disseminated. Sorry, spread beyond the primary site we call a disseminated infection. And if many organs are infected, we call it a systemic infection. Now, spreading beyond the primary site is not easy. Both physical and immune barriers need to be breached. What do I mean by that? Well, for example, this basement membrane is a physical barrier. Viruses, even if they're released basolaterally, the bottom of the cell, they will not go anywhere unless the basement membrane is somehow compromised. And of course, an immune barrier would be the presence of immune cells, antibodies, and so forth. Unfortunately, we ourselves compromise our own basement membrane in a variety of ways. Immune cells get through it all the time. And so where that immune cell, this macrophage is squeezing in, viruses can squeeze out. But also inflammation causes a breakdown of the basement membrane. What is inflammation? Well, it's triggered in many ways, but one way is by virus infection. Inflammation a virus reproducing in the cell produces cytokines, and that attracts other immune cells which produce more cytokines, and that breaks down the basement membrane. Why? In part so the immune cells can get past it and do some work on the infected cells. Now, breaking down the basement membrane isn't enough. The virus, to spread, needs to be able to come out of the basolateral domain, the bottom layer of the epithelial cells. For example, here are 3 electron micrographs of epithelial cells infected with 3 different viruses. Apical release means the virus is only released by budding, in this case, influenza virus, from the top of these epithelial cells. So here's influenza virus, which is really good at spreading in respiratory droplets, right? Because it buds from the top of the cells. If it budded from the bottom, it wouldn't be able to spread by respiratory droplets if it budded from the basolateral domain. Basolateral spread, here's measles virus budding from the apical domain. Also, poliovirus buds from the apical domain. Basolateral release allows virus to potentially have access to all the underlying tissues and eventually get elsewhere, as we'll see the mechanisms of in a moment. Here's vesicular stomatitis budding from the basolateral domain. So to illustrate, of course, if you bud from the basolateral domain, if there's no inflammation and breakdown of the basement membrane, you're not going to spread. So you need to have 2 things. You need to have basolateral production of virus particles and then breakdown of the basement membrane. So I want to tell you a story of Sendai virus to illustrate this principle. Sendai virus is a paramyxovirus. What you may say is a paramyxovirus, it's a family which includes measles virus. Okay? So this is a virus, yes, isolated in Sendai, Japan. They didn't mind having a virus named after them, probably because it just infects mice. It's a mouse virus, and it's used as a model in the laboratory quite frequently. Sendai virus can infect mice. You can put the virus in their nose. The virus will infect the respiratory tract. The virus buds apically and can be shed in respiratory secretions, but it doesn't cause systemic infections in the mouse, it is not dispersed beyond the respiratory tract. Now, investigators years ago made a single amino acid change in the spike glycoprotein of Sendai virus, which changed its properties. This allowed the virus to bud from the basolateral domain. So this mutant virus, when you infect mice with it, the virus spreads and causes a systemic lethal infection. So just one amino acid change allowing the virus to bud basolaterally is all you need in order to have the virus spread systemically. Viruses, once they get below the epithelial sheet through the basement membrane, they can then get into the circulation, the blood vessels, and spread in the blood, which we call hematogenous spread. All right? Practice that. Hematogenous. That's the way you say it. That's where the accents are. Now, in these sub-epithelial tissues, there are capillaries, tiny blood vessels, right? And there are also lymph capillaries. Lymph capillaries are also small vessels of the lymph system, which eventually feed back through lymph nodes and back into the circulation. So they're all connected. And it turns out that lymphatic capillaries are more permeable than blood capillaries. So viruses tend to enter them. They go through lymph nodes, eventually get into the circulation, and then they become hematogenous, and they can spread in the blood. Now, the presence of virus in the blood is called a viremia. Viremia, presence of virus in the blood, okay? And this is an experiment where we have made a viremia in mice by injecting virus into the vein, a tail vein typically of the mouse, and we're now measuring virus by infectivity, days after infection, and here's the virus titer, relative virus titer. So we put the virus in the blood on day 0, and if you take a time point right away, you see there's a lot of virus in the blood. Why? Because it's what you put in. It's what you inoculated. That's called passive viremia. And then after a day or so, the titer in the blood is much lower. It's largely entered various tissues. We're very good at clearing particles from the blood that goes through the liver, and it's filtered out very well. However, this virus clearly infected some cells because by day 2 now we see an increase in the virus, which is caused by reproduction of the virus in some tissue in the mouse. And that we would call a primary viremia. This then allows the virus to spread to other tissues. It may decline, as you see here, and then it could seed multiple tissues, and then you have a very, very substantial secondary viremia as a consequence. And this is a typical pattern for viruses that spread through the blood. Now, here is the pathogenesis of a virus infection in mice called mousepox. Again, it's a model for studying pox viruses in humans. It's a virus, it's a natural mouse virus that infects mice in the wild. And so the way the mice are inoculated, they're inoculated in the foot pad. All right, so you inoculate virus in the bottom here. And why do we do that? Because that's how these mice acquire infection in nature. They're walking around on the forest floor, which is rough, and they get little nicks in their foot pads. And if some virus has been shed by another mouse, or if they're intimate with another mouse, they're going to get infected in the foot pad. So you put the virus just below the epidermis, and the virus reproduces in cells in the skin. It enters the lymph and eventually the blood, and you have your primary viremia. This is 2 days after you inoculate them. The primary viremia leads to virus entering other organs, kidney, spleen, liver. I'm sorry, that's the spleen, not the kidney. The spleen and the liver, where the virus reproduces, causes damage, and those cells release virus into the blood. That's your secondary viremia that we saw in the previous curve, and that brings virus to the skin where it causes this rash. This is a mouse version of smallpox. And so you see the spread of this virus by two different stages of viremia. At the same time, the foot pad is swelling because that's where you inoculated the virus. And so this is inflammation caused by immune cells coming in. fluid coming in and cells coming in. Of course, viruses cause rashes in humans, like the rash that we've just seen in mice. And here are some examples of different kinds of rashes. We give names to the rashes because they are distinct. You can look at them and see how they differ and sometimes get an idea for the virus. Here, for example, is the measles virus rash, the smallpox rash, the chickenpox rash. And these rashes are given names like maculopapular. This is a maculopapular rash right here, where it's a flat rash with a raised center. Or there are vesicular rashes, where there's clearly a higher raising of the area full of fluid and cells. And so these are characteristic for different viruses. So these are some viruses of humans that cause rashes. Measles, of course, but Coxsackie viruses can cause rashes, particularly in kids. Parvoviruses. Erythema infectiosum. It sounds like a Harry Potter curse or whatever you call those things. Not curses, right? German measles, rubella virus, varicella zoster, the rash, chickenpox, or later on shingles. And Zika virus also may cause a rash as well. And typically the rashes are, viruses are at those sites in the skin, and then the immune system is responding, and that's what's causing the rash. Time for another quiz. Which of the following assist in viral dissemination in the infected animal? Viremia, basolateral release from epithelial cells, movement through the lymphatic system, inflammation at the basement membrane, and all of the above. And then while you're looking at that, let's check out some questions. Thank you, Jeremy, for your contribution. Yeah, play with it. It's fine. You know, stick with us. You'll see it evolve, right? I'm not wedded to being against this wall. This is a cool room. I might move it somewhere else. Aren't pathogens supposed to be killed by immune cells in the lymph nodes? No. What happens in the lymph nodes is that the T cells look at the antigens that are brought in by antigen-presenting cells like macrophages and dendritic cells, and they determine if it's foreign or not. And if they're foreign, they initiate an immune response. And then the effectors of the immune response, the antibodies and the T cells, go out into the blood and find the initial infection, and there they destroy it, not in the lymph node. Soaps affect skin defense adversely. So your skin, you know, it's a beautiful thing, your skin. It's got a microbiome. It's got bacteria that are resident and that you should have. So if you wash your hands too much, well, the palms of your hands, I suppose, don't matter. But the rest of your skin, I mean, showering once a day, I suppose, is fine because the microbiome isn't eliminated. But that microbiome produces antiviral peptides, actually, and antibacterial peptides that are among the first lines of defense. So not only is the skin dead, but there are peptides there that are antimicrobial. Very interesting. We owe so much to mice. We certainly do. Yeah. Do I agree with Paul Offit about not suppressing fever during infection? So what is fever? In part, it's thought to eliminate some pathogens from the high temperature, right? I don't think it eliminates all of them. We have not really ever had that practice, right? The response of a physician is to control fever, because if it gets too high, that can cause shock, right? My daughter years ago, when she was a baby, had a, what is it called when you have a high fever for a while and you get a seizure? She had a seizure from the fever. So you want to control it, right? So I'm not sure in every virus infection that's a good thing, but it's one of the things they do. If you go to a hospital and you have a fever, they'll try and control it. So I don't know what would happen if you didn't. I'm not sure that you would resolve any quicker. Acute virus infections are very rapid anyway, right? They don't necessarily determine if it's foreign or not in the lymph node, but that is a main site of determining whether it's foreign. That's where the antigen-presenting cells present the T cells. I'm sorry, PK Madness, that's a major part of determining that it's foreign. You can ask Brianna if you don't believe me. Yeah, we control fever because it can damage the brain and other, especially high fever, right? But I don't know about mild fever. Okay, let's go back to quiz. Febrile seizure. Yes, thank you. The Rolodex is going slower, as Dixon would say. Okay, what's the finding here? All of the above. That's right. Viremia, basolateral release, movement through the lymphatic system, inflammation at the basement membrane. They all assist in viral dissemination in the infected animal. Viruses can spread in nerves. Unsurprisingly, this is called neural spread. Viruses can, for example, enter the nerve junction in the muscle. As you know, your muscles are innervated. They have sensory nerves, but also motor nerves so that you can move them. And there's the junction of the nerve with the muscle there where viruses can enter. They can then move in the nerve into the spinal cord and even up into the brain because it's all wired together. This motor neuron in the spinal cord is wired to a neuron in the brain. So many viruses can enter at neuromuscular junctions, even from nerves in the skin, and enter the CNS. Now, what causes movement in nerves? Well, there's a series of motor proteins, of course, along the microtubules. Here's a diagram of a nerve with the cell body and a terminus separated by an axon in this case, or a dendrite. And within the nerve body, or the process here, there are microtubules with motor proteins that transport material towards the cell body or away from the cell body. And here's a herpes virus that is being carried in a vesicle from the nerve terminus up to the cell body. Many investigators have used this to map circuits in the brain. Here, for example, is an experiment where this is a herpes virus. Is actually pseudorabies virus. It's a virus of swine, and it's a herpes virus. It could be experimentally manipulated very nicely in the laboratory. And in this case, the investigators put the gene encoding green fluorescent protein into the pseudorabies virus, and then they stereotactically injected it into the rat brain. What does that mean? You put the needle, a very small needle, into a very precise position in the brain that you would like to know. How is this connected to another place in the brain? And then with time, you can see the virus spreading from nerve to nerve. So not only do viruses spread within the nerve, but they go from nerve to nerve. They're released in the cell body, for example, and then they infect another synapse, and they go throughout the brain. You can see here the virus moved throughout the brain. You can map the circuits by tracing this GFP. It's really brilliant. Now, when we talk about infections of the central nervous system, viruses being brought to the central nervous system, the brain and the spinal cord, that's the central nervous system, as opposed to the peripheral nervous systems, neurons outside of the brain and spinal nervous system, Bruce Carter. When we talk about CNS infections, these are the words we use, okay? And many people misuse them, even virologists. Neurotropic. A neurotropic virus infects neural cells. That's it. That's all it means. Doesn't matter how the virus got there, what it does. All it means is that the virus infects neural cells. A neuroinvasive virus can enter the central nervous system from elsewhere. So a dog biting you on the hand, giving you rabies virus, that virus ends up in your brain. That is neuroinvasion. Nothing else implied. Could be the end, but if it gets in there, it's neuroinvasive. Neurovirulent means the virus causes disease of nervous tissue. So that is not implied by the other two at all. Neurotropic, just reproduction, neuroinvasion, invading. neurovirulence. So how is this used? Herpes simplex virus has low neuroinvasiveness. When you get a cold sore, the virus tends to stay outside of the CNS. It will go into peripheral ganglia, but not the CNS. However, occasionally it does get in the CNS. And when it gets there, it has high neurovirulence. So it's obviously neurotropic because it's reproducing in neurocells and it's damaging them. So it has high neurovirulence. So herpes simplex reproduction in the brain is rare, but can be devastating. Mumps virus is highly neuroinvasive. You know, mumps used to be a common childhood pathogen. I got mumps as a kid because it was before the vaccine. I was a kid before the vaccine. And it turns out that in most kids, the virus got in their brains, but didn't do anything, didn't cause any damage. So high neuroinvasiveness, low neurovirulence. And then finally, the mother of all viruses, rabies virus. Why do I call it the mother? Not because it's the ancestor, because if you're not vaccinated, 100% lethality after getting infected with rabies virus. High neuroinvasiveness, high neurovirulence. So you get bitten on the hand by a rabid dog, the virus will most certainly get into your CNS. And once there, if you're not vaccinated, you'll probably die. I think that's quite a virus. Let's talk about how viruses then invade tissues. We've talked about viruses spreading in the blood, right? Now, how does it get out of the blood into tissues where it's going to reproduce? Here we have 3 scenarios for that. We have this scenario on the left, which is in a sinusoid. So this is the situation in the liver, the spleen, the bone marrow. The blood vessels, you barely could call them a blood vessel. It's just a collection of endothelial cells here, loosely joined with the cells underneath. These could be liver cells, for example. So it's just like a sinus, and the blood is flowing through this. So obviously, very easy for viruses to get through these holes in the endothelial cells. Now, in the other tissues, kidney, pancreas, ileum, colon, we have a better situation. We have a basement membrane, first of all, which is good, prevent virus from just going in willy-nilly. And the endothelial cells are joined, but there are pores. So not impossible to get across, but a little higher barrier than the liver. And then finally, we have the situation at the right, which we find in central nervous system, skeletal muscle, lungs, etc. These are proper capillaries where the endothelial cell is now one cell with a very tight junction. There's the nucleus. Because a series of endothelial cells make up the capillaries, make up the blood vessels. We have a basement membrane. So very difficult for viruses to get across this. So does that mean it doesn't happen? No, of course not. It does happen, although it's tough. How does it happen? Well, here are some ways that viruses can get across from the blood into underlying tissues. So here's our capillary. There's the lumen. There's the endothelium. All the cells join together to make a tube, right? Outside of it, we have our basement membrane. So here we have some virus particles floating around in the blood. They could, for example, infect the endothelial cells and be released on the other side. The infection could cause inflammation of the basement membrane and breakdown. They could be taken up and transcytosed across the endothelium, or they could be carried in lymphocytes or monocytes. These cells have the ability to get through the tight junctions of capillaries. It's a process called diapedesis. I like that word. It means cells getting through the tight junctions, and if they have viruses in them, obviously they're going to bring them into the surrounding tissues. And if this is the brain, that's how viruses get in your brain. So here's a summary of virus entry into the central nervous system. Here's the high-level view here, the brain proper, or parenchyma. We have our spinal cord coming in there. And so we have, the cerebral spinal fluid in the center. This is an area that has blood vessels. It's called the choroid plexus, the area where the cerebral spinal fluid is made. Those are loosely joined endothelial cells, and so many viruses can go into the CSF from the choroid plexus and then infect the brain proper. Brain, of course, the cerebrum has blood vessels in it, and sometimes viruses get through them by the mechanisms we just talked about. The meninges, of course, also has blood vessels, and viruses can get through them as well. Now, here's a close-up view of a blood vessel in the brain, showing you the tight junctions. Basement membrane isn't shown, is labeled, but I guess it's that gray here, okay? And then outside of it, we have astrocytes. Astrocytes, the major cell in the central nervous system, and as you can see, they are protective of these capillaries. But many viruses have acquired the ability to reproduce in the astrocyte, and that's one means that they get across. And we talked about one of those papers on a TWIV not too long ago. All right, let's get back to transmission of infection. We talked about measuring it by R naught and RE and RT and so forth. But once we have gone through this reproduction in a host, which we've just talked about up until the tissues, the virus then has to find a new host. So it has to be transmitted. The transmission is the spread of infection from one host to another. And of course, you need to do that to maintain a chain of infection. It's influenced by the virus and by the host, and that's why the R naught is a combination of both. And so a big question this past year is, are SARS-CoV-2 variants of concern intrinsically more transmissible? Has anyone done the right experiments to show that? In my opinion, no. It's all based on observation of movement of virus in the population. The human factor is often not considered in determining these RE, RT. I believe, and many others agree with me, that the variants of concern have greater fitness, and that's why they displace previous variants, right? The alpha variant arising initially in the UK, subsequently displaced by the delta. Why? The delta is more fit. So what is fitness? So it's something that is often confused with transmission. I would say think about Darwin's survival of the fittest, right? The finches whose beaks changed depending on what the food was on the islands. The fittest birds with the beak suitable for the food survived. The same thing applies to viruses. And viral fitness, so populations, host populations select viruses that are more fit. And what is fitness? Well, better transmission could be part of it, but it also could be levels of reproduction, stability of particles, immune evasion, many different properties. And so what we can say is that the variants of concern of SARS-CoV-2 are more fit, for sure. They displace previous variants, but why they're more fit, no one has figured out yet. The epidemiologists cannot do that. That's up to the virologists working in the laboratory, and people are doing this. We covered a paper not too long ago about the Delta variant, indicating that it seems to reproduce more quickly in cells, which would be a reflection of increased fitness, and that would drive it through a population. I want to compare this to influenza virus. Every year, antigenic variants of that virus arise that are more fit. They displace previous variants, and sometimes we have to change the vaccine to accommodate it. No influenza virologist calls them more transmissible. They are more fit. Now, when we talk about transmission from host to host of a virus, there are two general patterns. We can do animal to animal, human to human, for example. Or we could have a vector involved. Those are the two general patterns. So here, a human is infected with a virus, sheds it in the feces, on the skin, via respiratory droplets. It reaches another human, they're infected. That's animal to animal. But some viruses are not spread directly from human to human. They may be spread, say, by mosquito bites. Here, this particular mosquito, as we've talked about, taking a blood meal can spread viruses. And so here's an example of that with a rodent virus, a virus that infects rodents, which is transmitted from rodent to rodent, say, by a tick. The tick takes a blood meal, picks up the virus, infects another rodent. And in the wild, these interactions occur all the time. So animal-animal or animal-vector-animal, two general patterns of transmission. And for transmission, we also have terms that you will see you should know what they mean. We talk about horizontal transmission between members of the same species. If I give you my virus, it's being spread by horizontal transmission. If it is a different species, we call it zoonotic transmission. Vertical is between mother and child. It's a very special interaction, mother and child. It could be across the placenta or perinatally. Iatrogenic is when the activity of a healthcare worker leads to an infection. And nosocomial is when said infection occurs when you are in the hospital or a healthcare facility. And finally, we have germline transmission, which we know about. The agent's transmitted as part of the genome, and proviral DNA should ring a bell there for you. So here's a nice picture, and I used sheep here to try and involve more of the mammals than humans. So we have horizontal transmission from sheep to sheep, and then mother to child is our vertical transmission. Now, in many cases, but not all, shedding is needed for transmission. What is shedding? I think it's obvious, but it is release of virus from the infected host in some way. So we talk about respiratory secretions or respiratory droplets that are produced when you talk or sneeze, mucosal shedding, mucosal surfaces. Your genital tract, for example, has virus. It can infect someone else. You can shed virus in urine, semen, and feces. Virus can be in skin lesions. That's all shedding because the virus is effectively leaving you as part of the infection, and it intercepts someone else. Now, blood doesn't necessarily have to involve shedding, right? If you get a contaminated pint of blood given to you during surgery and you get the virus, it's not being shed from the infected host. They've taken the blood. I know it's a technical detail, but shedding, just imagine shedding, sneezing, or defecating and releasing viruses, right? And as I say here, shedding is not always needed for transmission, the blood supply. And the blood, of course, could involve shedding if you were cut and you bleed and someone else gets infected. This is the way we think HIV began, when a hunter caught a non-human primate and was butchering it and cut himself or herself, and the blood from the animal went into his or her wounds. So that could be shedding. So when we don't need shedding, blood supply, insect vectors, there's no shedding involved. Germline, of course, and vertical involving transplacental because it's not really shed, but I would say perinatal is probably shedding because that blood is coming out. Obviously, there are fine lines here, but I think you get the picture. So here's a picture of virus shedding from the respiratory tract. This young lady sneezing, and they put a light behind so you can see the droplets, and you can see they're really big ones that fall to the ground, and then they're different size droplets. And in fact, people study these extensively, and they are categorized into droplet sizes: large infectious droplets, which fall to the ground quickly, smaller infectious droplets, and then the smallest, the infectious droplet nuclei, which can go up to 160 feet and more. They're very small, so you need to have a very infectious virus or a lot of virus, and the small droplet makes it hard for a lot of viruses to be transmitted in that route. And so as you expel droplets, they tend to evaporate depending on the humidity and get smaller and may travel farther, in fact, and can still contain the virus. So for SARS-CoV-2, we say stay 6 feet away because we think mainly these small infectious droplets are involved in transmission. But there are obviously some cases where finer droplets are involved. The fraction that represents is not clear to me at all. And these secretions can be made by coughing, sneezing, and speaking. Also, nasal secretions, mucus that you contaminate public appliances with, subway poles and door handles and so forth. Some viruses are effectively transmitted that way, in particular rhinoviruses. One of the best examples is years ago they did human transmission studies with rhinoviruses where they took one person who had a common cold and they put them in a room with 3 others and they played cards. And the virus was transmitted on the cards. more effectively than by aerosol. Anyway, there's a good video here where they did a slow motion movie of several people sneezing. It's quite gross, but the students seem to like it. This is a cool experiment that we covered on an old TWIV where they recruited 156 individuals in a college community. All these individuals had confirmed influenza. They wanted to know, how is it transmitted? And they put them in front of this contraption, which is called the Gesundheit 2. I guess there was a 1. And this is the improved model. And they sit in front for a fixed amount of time, and they go through exercises where they're just breathing. Then they go through, they'll recite the alphabet and so forth. And every letter has a different characteristic, plosive, right? So, and then they collect what's in this tube, and they look for a virus by PCR. And what they found is that infectious virus shedding, actually they also looked by infectivity, is in fine aerosols produced by breathing and speaking. So just breathing and speaking is enough to deposit virus-containing aerosols. Sneezing doesn't make an important contribution to virus shedding more than just talking or breathing, and coughing isn't necessary either. Isn't that something? I mean, this is influenza, so we don't know how general it is, but I find that fascinating. Now, here is a comparison of 3 different viruses in terms of shedding and transmissibility. So let's take a look to see what general principles we can get. So first, let's look at SARS-1, 2003. And what we have here, we have a graph showing virus shedding. And here's days from symptom onset. So symptom onset here is the red line, right? So you have a incubation period, which is the blue. Then you have symptom onset. And then this is a map of shedding, a graph of shedding. So this incubation period is 4 to 5 days for SARS-1. And the peak of shedding, which would mean that's when you're maximally transmissible, right, occurs about 10 days after symptom onset. And for SARS-1, most of those people were in the hospital because it made you very sick. And so that's why it was easy to contain SARS-1, because people were in the hospital. Also, you see, there was no shedding in the pre-symptomatic phase. So no asymptomatic shedding. That's why SARS-1 is gone. Seasonal influenza is different. You can see that there is shedding about 2 days before symptom onset, then it continues afterwards. So you have a period of time when you can transmit in the absence of symptoms, so you're walking around the community. And finally, SARS-CoV-2 incubation period could be 1 to 14 days, an average of 5. Here's symptom onset, which is roughly at the peak of virus shedding. So there is a period here during the incubation period where you're transmissible. So that's the problem with SARS-CoV-2 and seasonal influenza, that you can be shedding and be asymptomatic, so you don't know that you're infecting other people. So it's very clear now that the virus is transmitted during incubation period and from asymptomatically infected persons. And this was debated at the beginning of the outbreak, and it was, I think, driven by politics, because the science clearly shows this to be the case. All right, we have one more question today. Which statement about viral transmission is not correct? A, all virus infections are transmitted by shedding. B, the route is determined by the site of virus shedding. C, transmission is required to maintain a chain of infection. D, speaking can produce an aerosol that can transmit infection. And E, horizontal transmission is among members of one species. All right, check that out. We'll go to some questions here. Okay. Yes, we have microbiome in the skin on top protecting us. You bet. Last week it was very high fever, 107. Wow. Yeah, you'd want to treat that, right? So it's not suppressing fever isn't suppressing the immune response. Well, I guess it depends what you're doing, right? So if you targeted a specific cytokine, that would just be part of it. Yeah. So basically, if you're targeting what cytokine causes fever, and that's the basis for fever, then yes, you could suppress the immune response. Yep. Yep. No, self-reactivity occurs in the thymus and bone marrow, absolutely. And all the self-reactive T cells are gone. Right. But we were not talking about self-reactivity. We were talking about detection of foreign antigens. Yep. Yeah, it's what I give my regular class. And what I get, what they will get in the spring when I teach the regular class in person, yeah. Can we say that a neuroinvasive virus is also neurotropic? So it depends how you detect neuroinvasion, right? If you're detecting it by reproduction, then yes. But if you aren't, if you're just, say, looking by PCR to see if the virus can get in, then technically it couldn't. But I know in the old days we didn't have that, so I would say most of the time, yes. Yep. Regarding rabies, I saw a study that suggests some people are asymptomatic based on immunological studies of tribal areas of Peru. So the problem there is that you don't know if it's a virus that causes cross-reactivity. So it's not rabies, but a similar virus. Same thing has occurred with Ebola viruses, right? It's not clear that it's Ebola virus or something related that doesn't cause the same disease. But it's an important question, yeah. So here we have an analysis of diapedesis from the Greek, act of leaping through. Very good, thank you. Do we know of any viruses that change host behavior to increase the R naught? Make you more gregarious? I'm not aware of any. I'm only aware of the viruses that make you sick, so you stay home or you go to a hospital, right? Is there a specific definition of fitness? It's the ability of a virus to reproduce in the host population. But what causes it can be multifactorial. Would viruses from the quasi-species be under different evolutionary pressures between different tissue cells within the same individual? Absolutely. Absolutely. You can imagine different pressures in different cells, different environments, different receptors, different immunity. Yeah, for sure. Severe COVID tends to be the host response. I always hear viruses evolve to be more transmissible and less virulent, even though we have no human data for that. Okay, there's very little data. We're going to get to that in another session. Is it viruses evolve this way or change the immune response? I think, look, we don't know in humans, but there's an animal experiment done in Australia where the animal changed as well. And let's save that for evolution, okay? Is vertical always birth related? Mother to child, but not father to child, just mother to child. So I think it could be mother to child early in life by other means as well. Does decreased fitness mean less deadly, more compatible with the host? Increased fitness? It could. It could. It depends on what's mediating. So you can, I think, polio is a very well-adapted pathogen because it mostly doesn't cause disease, and it's very fit in the host. And we don't know how it got there, but that's one road that it could have gotten there, yes. How is poliovirus transmitted? So you ingest fecally contaminated material, and it goes into your intestine, reproduces there, and then you shed it in the feces. Fecal-oral contamination. People don't wash their hands, especially kids. They get poop on their hands. They play with other kids. They touch objects. The poop gets on them. Happens all the time. That's how noroviruses are transmitted that cause gastroenteritis. We'll talk about that. We will. Low exposure to viruses that we are already immune to keep our immune system well trained. It will, although memory does not require exposure to the pathogen, but it will boost it if you are. Yes, for sure. can you get infected by SARS-CoV-2 if you touch a door handle and touch your mouth? I think it depends how much virus there is. Certainly if you touch the nose, then you would have a direct route to the nasal mucosa, right? But yes, mouth may degrade it. But if you've got a lot of virus on your finger, I think that's very unlikely that there would be so much. I mean, there would have to be a visible deposit of mucus on the door handle for you to do that. So I think it's highly unlikely. Are nerve endings specially protected in some form? Since the blood-brain barrier is such a special thing, you'd hope for something like that. Yeah, they have a myelin sheath, right? Or maybe that's just the nerve body. But, you know, not all receptors for viruses are at nerve endings. I think that's part of it, and not all viruses will reach them. So yes, what's the book above virology? That's... This is the subject of the last TWIV, A Shot to Save the World, the inside story. This is really good, and we interviewed the author on the last TWIV. I thought it was a good place to put it. And the other is Fields Virology, latest edition. So plant viruses do very cool things to plants, yes. They make the plants produce odorants that attract vectors and so forth. Very cool things happening there, yeah. Yes, there's a caterpillar virus that makes it climb to the top of the tree and explode. There are viruses that make ants go to the top and they get eaten by birds. Yeah, they do all kinds of, but not in people, yeah. Viruses with long incubation periods like rabies. Yes, you can be vaccinated in time to prevent the neuroinvasion. That's right. And the further the bite is away from the CNS, the more time you have. But that's the usual response to a rabies bite. You get monoclonal antibodies injected at the bite site to neutralize virus, and then you get vaccinated. And that will protect you from neuroinvasion. Yep. Passive active vaccination. That's right. What property of enteric viruses allows them to survive the acidic environment of the GI tract? Oh, the capsids are stable. So, you know, poliovirus, like this one. So this is poliovirus. So the thing about having viruses on my shelf, I can pull them off and show them to you, right? I'm going to fill this shelf with viruses. They're very stable capsids. They don't come apart at low pH. But even envelope viruses can be stable. So many enteric coronaviruses that infect non-human animals are also stable. They must have a very stable envelope conferred by viral proteins in part. Could the virus adapt to a new cell inside the infected person? It could. You know, I don't see evidence of that, but certainly in animals, viruses are known to change tropism, in particular some coronas. So, yes, there is evidence for that. Yep. Would droplets of SARS-CoV-2 which fell to the ground and dried later raised and be breathed in? I think they'd be inactivated. They're not going to last very long in a dried droplet, maybe a few hours at the most. But again, it depends how much virus is there. So if the virus is more dangerous in the nose, would it be more important to cover just your nose than just your mouth? No, because you're exhaling from the mouth, right? Some people do. And I suppose if you inhale through your mouth, it's going to go back to the tract. So the other scenario was you told me if you put a contaminated finger on your mouth, but if you actually inhale through your mouth, you can inhale virus. So you have to cover both, for sure. How do I think SARS-CoV-2 can cause GI infections given that it's envelope? That's a good question. Nobody understands. It must be that the membrane proteins give the envelope stability because influenza virus will not survive the human GI tract. but we don't know. It's a good question. Double glove with PPE if I'm going to touch the viruses. Yeah, right. All right, let's check out our quiz here. Okay, most of you got the answer. A, all virus infections are transmitted by shedding. Not all are transmitted by shedding, right? Some of them vectors, germline, etc. The route is determined by the site of shedding, for sure. Transmission is required for a chain of infection. Speaking can produce an aerosol. Horizontal is among members of one species. Those are all correct. All right, let's wrap this up. Some at the end here, some other influences on virus infection. Geography is a big one. It can restrict the presence of the virus because the virus may be transmitted by a specific vector or an animal reservoir. An example of this, chikungunya virus, how the vector can affect localization of virus infection. And so this is a virus transmitted by mosquitoes. And currently it can be transmitted by two mosquitoes, Aedes aegypti and Aedes albopictus. And these are the distributions of those mosquitoes in the U.S. So chikungunya is a toga virus within the alpha virus genus. It's an envelope virus, spike glycoproteins beneath a nucleocapsid with plus-stranded RNA. It's spread historically by Aedes aegypti. You get infected, you get rash, you get fever, you get joint pains. It causes an arthritis-like illness. Originally found mainly in Africa and Asia, never the Europe, never the U.S. However, in 2004, the virus began to spread initially from Kenya to India, and then the first outbreak in Italy in 2007. And this a major outbreak of chick was on the island of Réunion early on, where it had never been seen before, and as it's a French island, many of those travelers brought the virus back to Europe. So why did it change? Why did it go from blue to these yellow countries? Well, these recent outbreaks are associated with a new vector, Aedes albopictus, and the virus was selected to have a one amino acid change in the viral glycoprotein, which is called E1. One of the two is E1. And that was enough to get higher reproduction in the mosquito, and then the mosquito became a competent vector, whereas previously Aedes aegypti only, and that is more geographically restricted. Aedes albopictus is more extensive in the U.S. It was actually introduced into the U.S. In the 80s by the used tire trade. It came into the U.S. in Houston and has since been spreading northwards. And so the potential is that this virus could be in the U.S. as well. We do have chick infections in the U.S. So far we've had 192. Well, in 2017, there were 192 imported cases into the mainland, all imported, no local transmission yet. There is some local transmission in Puerto Rico where they have Aedes aegypti, but in the U.S., here's another font issue that I need to fix because I can't have that looking like that. And these are all the states where the imported cases occurred. Now, here's the range of Aedes albopictus. So this is what you have to look at for having spread within the U.S. This is increasing because, as you know, global climate is increasing, and here's a modeling of the climate increase throughout Europe. And as the climate increases in temperature, the Aedes albopictus range will increase, and so eventually this virus will end up throughout Europe and the U.S. as well as a consequence. Now, many virus infections are seasonal. Here are some graphs that show that. Here's rubella in the U.S. from 1963 to 1968. You can see a summer prevalence. Here's influenza, which, of course, in temperate climates is a winter disease. And here's polio, which is seasonal, but which season depends on your latitude. And so in the U.S., polio is typically a summer disease, but in other countries, the summer is at different times of the year, obviously. So what causes seasonality? We pretty much don't know. Anyone who says they do is lying or misleading. There's one good experiment, in my opinion, about how seasonality influences influenza virus transmission. And this is an experiment done in guinea pigs, hamsters, where you can put hamsters in adjacent cages. You can infect one and have airflow from one cage to another, no contact, and the infected hamster will infect another. So you can look at transmission. And in this experiment, they're looking at the effect of temperature and relative humidity on percent transmission. So, for example, if you increase the temperature, that's in red here, at higher temperature, transmission drops because the virus particles are unstable. Now, it's interesting that if you continue, I'm sorry, that's the wrong, let me start over. So we have two different experiments, 5 degrees Celsius and 20 degrees Celsius. So transmission is much better at 5 degrees Celsius overall than 20 degrees. At 20 degrees, the virus particles are inactivated. But as you see, if you increase the relative humidity, look at 5 degrees first. That's the clearest. The transmission drops even at 5 degrees. Why is that? Because the droplets are taking on water. Remember, you're sneezing or talking out droplets which contain virus. At increases in relative humidity, even at 5 degrees, the droplets are getting bigger. They're falling to the ground faster. That's the theory. And so they're not transmitting as well. So in the Northeast, under dry conditions, low temperature, this may explain why transmission is favored then. At higher temperatures, at some intermediate humidity, 50%, the transmission drops, but then it increases at increased humidity, even at higher temperatures. And it may involve the stabilization. So this could explain why in the tropics there are seasons of influenza, even though it's not low humidity and low temperature. Perhaps this explains it, or perhaps there are other modes of transmission. But essentially, low temperature, low humidity favors it in the Northeast, it seems. This may explain it. But all the other seasonalities, we have no clue. All right, now next time, which is Monday, we will start talking about immune defenses. We have 2 sessions on that next week. The first will be intrinsic and innate defenses. And let's see what we have here. In the meanwhile, for questions, thank you, Tom, for your support. Really appreciate it. Since we are always moving through a sea of viruses, and since viruses are always evolving, how come we get less routinely sick as we... So you're saying we get less sick than we used to be? Well, maybe medicine is better. I don't know. It's a good question. I mean, it may be that the onslaught of viruses and microbes has trained our immune system. I don't have a good answer. I'm sorry. By the way, thank all the mods today for helping out. Really appreciate it. Thank you, Kate, for your support. I have the midterm exam up in a separate window, ready to go. Go for it. Do it. Thank you, SARS, for your contribution. I'm going to get Vincent to admit that single-stranded plus RNA viruses are king. I already have. I told you they're the king. If I were to design a virus, that's what I would design, for sure. I agree. Oh, here are the mods. We had today Mr. Ozzycam. We had Les and Steph for modding. Thank you very much. Yes, the time-lapse maps models, right? But it's going to get very warm unless we do something, and it's really sad that we can't get it together to do something. Thank you, Barb Mack, for your support. You sure this isn't 80s Aegypti? No, I'm pretty sure that I got that as an 80s Albopictus. I will check. I'm sure you're good with your mosquitoes, though. That wasn't for me. Does humidity affect enveloped or capsid or envelope viruses differently? It might. I don't think we have enough studies on that at all. Do viruses and mosquito vectors reproduce in the mosquito? Yes. Most require reproduction in the mosquito. There are a few examples of mechanical transmission where there's no reproduction needed, but most do require reproduction. The virus goes into the gut. It infects cells lining the gut. It becomes systemic and ends up back in the salivary glands of the mosquito. Yeah. All right, it's 1 p.m., folks. Thanks again for coming. Have a great weekend. Be safe. And see you next time. Bye-bye.